Hello, folks. It is Mr. W, and I am really happy to be here to do some AP Bio review. And I see that there are already some people in the audience, and that is really fantastic. And I would like for people to just let me know, A, that they can hear me, and B, tell me where you are watching from. Where in the world are you getting ready to crush the AP Bio exam? So let me know about the towns, about the cities, about the villages, about the states, about the countries where you are preparing for a big challenge, and that is the test that you're going to be taking on May 5th. And thank you very much, Caitlin, for letting me know that you can hear me loud and clear. That is always so good to know. And Ruby, you're from Georgia. That's fantastic. New York City, Bakersfield, California, Glenside, Pennsylvania, Brantford. Where's Brantford? Anyhow, tell me what state Brantford is in, Booper Schmooper. And anyhow, I'm really glad to have you here for this review session. We're going to do some MCQs. We're going to do some FRQs. We're going to dive into it. And I just want to let you know how happy I am to have you here with me. And Victoria, thank you very much. Really great to hear that. I've been working hard to provide you with good materials to help you learn the greatest subject of all, biology. And I hope that it's just the beginning of your journey of learning this AP Bio test that you're going to be taking in just a couple of days. Let's do a little bit of adjustment. Yeah, that's looking good. And just remember that I've set up so many resources for you on my website, learn-biology.com. Don't forget that dash. That's where you want to be doing your studying. I'll tell you more about that as we get deeper into the show. But I think we should start with some questions. Let's start right away. So here's a question, and it's about the origin of life. So let's think about this. Here's what I'd like you to do. When I give you this question, I'd like you to enter in your answer. And then when I say send, I want you to all send it at the same time. Send it at the same time. And that way we'll have a cascade of answers. Everyone will have the opportunity to think and to, experience the question. Okay, so here's a question. By discharging electric sparks into a laboratory chamber atmosphere that consisted of water vapor, hydrogen gas, methane, and ammonia, Stanley Miller obtained data that showed that a number of organic molecules, including many amino acids, could be synthesized. Miller was attempting to model early Earth conditions. Put a different, oh man. I sometimes get a little bit trigger happy with my cursor. Anyhow, that didn't change your experience. Miller was attempting to model early Earth conditions as understood in the 1950s. The results of Miller's experiments best support which of the following hypothesis? A, the molecules essential to life today did not exist at the time Earth was formed. B, the molecules essential to life today could not have been carried to the primordial Earth by a comet or a meteorite. C, the molecules essential to life today could have formed under early Earth conditions. And D, the molecules essential to life today were initially self-replicating proteins that were synthesized approximately 4 billion years ago. Folks, this is a question from Unit 7. The topic is the origin of life. And I would like you to put into the chat an A, a B, a C, or a D. Go ahead, send it now. A, B, C, or D. Let me see it in the chat. Let's go. Let's go. I only see one response so far. The responses are coming in. Fantastic. Almost everybody is getting, in fact, every last person who's putting an answer in is getting this right. What a great group of students I am lucky enough to be with tonight. The answer indeed is C. And let's talk about this. The molecules essential to life today did not exist at the time the Earth was formed. This is how life came about. Life came about through the emergence of these Molecules. Molecules essential today could not have been carried to primordial Earth by a comet or a meteorite. Indeed, they could have. And the answer, of course, is C. And this is just wrong. And you know, so much of these multiple choice questions is about using a process of elimination. This experiment is the one that was carried out by Stanley Miller. Let me get this banner out of the way. There we go. So now you can see that what Miller did was he built this apparatus, and there was water that was boiled over here that set up a column of steam that went into this chamber over here. There were sparks that went through the atmosphere that Miller put in here. That was ammonia, things like ammonia, methane, hydrogen gas, and water vapor was in there too. There might have been some nitrogen gas. In any case, after running this for a period of time, Miller sampled the stuff that was accumulating down here, and he found that there were amino acids in the liquid. There weren't all 20 of them, but there were a couple of them. And that indicated that from non-life, the precursor molecules of life could emerge. And this is sort of the archetypal origin of life experiment. What's the basic idea? It's the model for many subsequent experiments, and it proved that under abiotic conditions, the monomers that make up the chemicals that make up living things today, including you and me, could be formed. A lot of text on the screen. Hope that's visible wherever you are. I will read it and hopefully that'll help. In a hypothetical population of beetles, there is a wide variety of color matching the range of coloration of the tree trunks on which the beetles hide from their predators. The graphs below illustrate 4 possible changes to the beetle population as a result of a change in the environment due to pollution that darkened the tree trunks. Which of the following includes the most likely change in the coloration of the beetle population after pollution and a correct rationale for the change? A, the coloration range shifted towards more light-colored beetles, as in diagram 1. All right, so here's the original population, this dotted line. They're suggesting that it shifted to the left, lighter color. The pollution helped the predators and the darkened tree trunks. B, the coloration in the population split into 2 extremes, as in population 2. Both the lighter colored and the darker colored beetles were able to hide on the darker tree trunks. C, the coloration range became narrower, as in diagram 3. The predator selected beetles of the color exchange. And 4, the coloration of the beetles shifted towards more darker colored beetles, as in diagram 4. The lighter colored beetles were found more easily by the predators than were the darker colored beetles. Ladies and gentlemen, right now, don't wait. Put in the, oh, did people already do it? People already did it. Did I say send? No, I didn't say send. Let's wait so that people get a chance to do this. And that will make this go better. And indeed, folks, the answer is D, as you can see over here. Now, this is a great question for a couple of reasons. One is that this question is helping you review types of selection. So diagram 1, diagram 4, those are both directional selection. And that's where the average phenotype in the population gets pushed in one direction or the other. In this case, because there's selection for darker colored beetles that would be able to camouflage themselves against the, I'm laughing because I got a super sticker. I've never gotten a super sticker before, so that's just kind of cracking me up. And Snickers, I'll answer your question in just a minute, but thank you so much. So here's what I want you to note. That is directional selection. This one in number 2, that is disruptive selection. And that is when there's selection against the average phenotype. And over here, this is called stabilizing selection. And it's when there's selection against the extremes and for the mean. This is the kind of thing that happens with human birth weight. It's bad for a baby to be too big because that can cause difficulties during childbirth. It's bad to be too small because small babies don't survive as well. So there's selection for babies that are right in the middle. So Snickers, 99-cent super sticker. I've never had that happen before. That is so awesome. Wow. I don't know what I'm going to say to that. Snickers, if you have a question, I'd be happy to answer it because, I've actually said that before, that if you use the super sticker thing, you get to answer your question. So Snickers, thanks a lot. And I really appreciate it. That is really cool. I got a super sticker for 99 cents. That's just cracking me up. Okay, great. So Snickers, you wanted to ask what? I'm looking at your question. Can I do a breakdown of how to answer the AP question if you're stuck? Yeah, okay, sure. Let me talk about that in a general way. Your best friend when you are doing these multiple choice questions is to ask yourself a couple of questions. Let's think about what a multiple choice question is. It's one true statement accompanied by 3 lies, right? That's how it works. One statement is true, the other 3 are lies. They're false, they're incorrect. So you want to ask yourself, is this true? Does it make sense? Stuff like that. So like, for example, if you look at question A, go like this. Question A, the coloration range shifted towards more light colored beetles as in diagram 1. Well, the trees got darker, so they wouldn't shift towards more light colored beetles as in diagram 1. The pollution helped the predators and the darkened tree trunks. So that one can't be true. So let's eliminate A, and that's what we're going to do. Now for B, the coloration split the population in 2 extremes. The lighter colored and darker, no, that wouldn't work in this case because we're going to push the population in one direction. So we're going to eliminate B. Let's look at C. The coloration range became narrower. The predators selected beetles at the color extremes. No, the predators couldn't see the darker beetles, so this wouldn't happen. And that leaves you with D. So Snickers, that is just one option. And just to elaborate on this a little bit, this is Darwin's theory of natural selection, which of course is the most important idea in biology. And basically what you have is the situation that we just looked at. You have inherited variation in this population of the beetles from recombination and mutation. You have a reproduction rate that exceeds survival rates. So many individuals are born, few survive. The survivors have beneficial traits. And in this case, what's happening is the beneficial trait is blending in with the environment. This is a visual predator. So any animal that sticks out is getting eaten. And the way I like to think about that is that means that its DNA is getting consumed and is being removed from the gene pool. It can't be passed on. Whereas the DNA in these better colored beetles is persisting. It's persisting because it's not being consumed. The survivors have beneficial traits, and mutation creates new variants. You repeat that over and over, you get adaptation. So that's how that works. And I just want to say thanks a lot, Super Snickers. That's really cool to have given me that banner. And if anybody else wants to do that, feel free. Thanks a lot. Really appreciate that. Didn't see somebody's super chat. Did somebody else do a super chat? Don't see any other super. Oh, wow. Holy cow. I do see another super chat. Okay. I really appreciate this. Can I get a shout out for my AP Bio teacher, Mr. Sue? Yes, Mr. Sue. I just want to tell you how much I appreciate you. You've been doing a great job teaching your students wherever you are, and they appreciate you. So, Mr. Sue, thank you so much. And I'd actually like to say, thank your AP Bio teacher because they've been working hard all year trying to guide you to the heights that you're at right now, the understanding that you're at. So thank you very much. And keep those super stickers coming. I really appreciate it. So let's go on with this quiz. The figure below shows, let me do that. The figure below, and I'm removing the super sticker just because it obscures the screen a little bit, but keep those super stickers coming. Thank you so much. The figure below shows several steps in the process of bacteriophage transduction in bacteria. Which of the following explains how genetic variation in a population of bacteria results from this process? Okay, got 4 choices. Don't send the answer until I say send, or else a lot of people's experience will be somewhat compromised. So bacterial proteins transferred from the donor bacterium by the phage to the recipient bacterium combine with genes on the recipient's chromosome. The recipient bacterium incorporates the transduced genetic material coding for phage proteins into its chromosome and synthesizes the corresponding proteins. The phage infection, the phage infection of the recipient bacterium and the introduction of DNA carried by the phage caused increased random point mutations of the bacterial chromosome. DNA, finally, this is choice D, DNA of the recipient bacterial chromosome undergoes recombination with DNA introduced by the phage from the donor bacterium, leading to a change in the recipient's genotype. So you got 4 choices, A, B, C, or D. I've read them. You send them now. A, B, C, or D. Let's see the answer coming through. Come on, folks. Send those answers, A, B, C, or D, to this question about bacteriophage. Okay. And I'm seeing a lot of D's come through, and that's filling me with joy, optimism, and hope, because D is indeed the answer. So basically what's happening is that when a phage infects bacterium, a phage is a kind of virus that infects bacterium, and I know that viruses are not a big part of the curriculum anymore. They used to be, and they should be, but here's a phage. It's injecting its DNA. And then what it does is it takes over the cell, and then it causes a cell to become kind of a zombie virus factory. And the phage are reassembled over here with pieces of genetic material that the victim cell's ribosomes, I'm saying this in all wrong ways, the phage infects its genetic material. It gets the cell to use its DNA polymerase to recreate that genetic material. It also gets the infected cell's ribosomes to create the viral proteins. Those recombine, and then there are new phages that are carried out. But here, a little mistake is made. And this phage is carrying some DNA from its host. It's carrying it to a new victim, injecting it in. And you see this is noted as S+ over here. That's going to recombine with the new cell's DNA. And so now what we've done is we've increased genetic information in the recipient. We've changed it. And this is one of the several ways in which bacteria, even though they clone themselves through reproduction, wind up having their genomes change over time. The others are things like mutation, conjugation, transduction, which is what we see here. So this is all Unit 6 stuff. You can learn more about it on learn-biology.com. I've got a section about it. And the comments are coming in pretty fast. And okay, nothing about this. All right, let's go on to another question. I will try not to. Okay, this is a tough question, but you just got to think about it, because like this stuff isn't exactly taught, but a lot of what AP biology is just being smart about interpreting diagrams. So I'm going to take a sip. And I want to just say something. Like, I'm seeing some comments, like this one by Legend Fire. I didn't study yet. Am I done for? Well, I'm looking at the date, and it's Monday, April 28th. And you've got many days. In fact, I'm going to get myself a little bit bigger to give you this little pep talk here. You've got many days to study. You've got hours and hours in front of you. but you have to start to be super efficient. And what you have to do is you have to use study techniques that enable you to find the gaps in your knowledge and then quickly fill them. That is not necessarily by watching videos. It's by doing what we're doing now. It's by taking lots of tests and seeing what you don't know. Because it's very easy to be overconfident in a study process, but you have to test yourself. And you should test yourself by lots of multiple choice questions, and you should test yourself by lots of FRQs. You should be doing what you're going to be tested on. You're not going to be tested on whether you can watch a video. You're going to be tested on whether you can answer multiple choice questions. You're going to be tested on whether you can write FRQs. And that's why what I want you to do is answer multiple choice questions, answer FRQs, and the place where you can do that is on learn-biology.com. So sign up. I've got hundreds of multiple choice questions. I've got hundreds of free response questions. And my free response questions are AI scored. So you'll get feedback on your free response questions, and you'll get the answers to the multiple choice questions. So there is time, but you have to set the time aside. And what is setting the time aside? It's not like saying, oh, I'm going to study. It's saying, after I finish this, watching this video with Mr. W, I'm going to study for 2 hours. I'm going to do that from 8 to 10 p.m. And then on Tuesday, I'm going to study from 7 to 10 p.m. And then on Wednesday, I'm going to study from 7 to 11. And I'm going to keep on doing that for hours every day. So it's up to you. If you put the time in and you study, you can do it. But if you're like, oh, I'm going to put in a half an hour every single day between now and the AP Bio exam, then I can't really do too much for you, even with the resources that I have for you at learn-biology.com. Okay, so I'm going to make myself a little smaller now. We're going to go back to doing some hard multiple choice questions. And this is the model for the kind of studying that you should be doing and that you can do on learn-biology.com. Okay, the first diagram to the right, this one over here, shows the levels of mRNA from two different genes, bicoid and caudal. Now, you don't know what those are. That's the whole point. At different positions along the anterior posterior axis of a Drosophila egg immediately before fertilization. You might not know what any of that means, but you know that this says anterior, this says posterior. Anterior means front, posterior means back. The second diagram shows the levels of the two corresponding proteins along the anterior posterior axis shortly after fertilization. So this is mRNA and this is protein. And we know that mRNA codes for protein. Why? Because we're in AP biology and we've learned that DNA makes RNA, makes protein. So knowing that, let's see if we can figure out this question. Which of the following conclusions is best supported by the data? Bicoid protein inhibits translation of caudal mRNA. So here's bicoid protein inhibits translation of bicoid mRNA. I don't know about that. I'm seeing that this bicoid mRNA is being produced in the front, and this caudal mRNA is staying constant over time. Interesting. Because, well, let's look at the other choices. Bicoid protein stabilizes caudal mRNA. Here's bicoid protein, caudal mRNA. That doesn't look right. Translation of bicoid mRNA produces caudal protein. Caudal protein stimulates development of anterior structures. Folks, I want you to use your biology smarts and put in an A, B, C, or D into the answer. And I see somebody put A or B. Don't do that on the AP Bio test. Of course, you can't. So what's the answer here? The answer is A, which actually, let's think about this. Bicoid protein inhibits the translation of caudal mRNA. Yeah, it's only when bicoid protein goes down that caudal protein goes up. So it's just about looking for relationships, even when you don't know what the hell they're talking about. Excuse me for slightly swearing, but that's really how this goes. All right, you're looking for what makes sense. You're looking for the obviously false answer. So, like for example, you could eliminate B right away. There's no evidence for that. You could eliminate C because that doesn't make sense. Translation of bicoid mRNA produces caudal protein. There's a bicoid mRNA, there's a bicoid protein. So you can eliminate two of these right away, which means that you're stuck between. In fact, you can eliminate D also because as caudal protein goes up, it's all in the posterior part. So just by a process of elimination, you could get to answer A, even if you didn't understand what was going on. Love this question. The vertebrate forelimb initially develops in the embryo as a solid mass of tissue. As development progresses, the solid mass near the end of the forelimb is remodeled into individual digits. Which of the following best explains the role of apoptosis in remodeling of the forelimb? The reason why I'm laughing is because I got this really nasty comment on YouTube where somebody went on and they did all this research into Greek, and the way that you actually pronounce this word is apoptosis. But I don't know why they needed to be mean about it. But anyhow, I've tended to say apoptosis, but I don't know. It doesn't matter. Apoptosis, it's about, well, I don't want to tell you what it's about. It's really sort of a biology term that you should know. I'll tell you about it in a minute. Apoptosis, I'm going to say it that way, replaces old cells with new ones that are less likely to contain mutations. B, don't send yet, guys. Do not send, all right? Do not send. Apoptosis involves the regulated activation of proteins in specific cells of the developing forelimb that leads to the death of those cells. C, apoptosis involves the destruction of extra cells in the developing forelimb, which provides nutrients for phagocytic cells. D, apoptosis in the developing forelimb triggers the differentiation of cells whose fate was not already determined. Folks, send it now. Let's see those answers stream in. And just like what Cheapy said, don't send until I say send. Okay, let's see those answers streaming in. Beautiful. Good thing we're doing this because a lot of folks are getting this a little bit wrong. The answer is B. And here's what apoptosis is. Apoptosis is programmed cell death. It's when genes get expressed that basically tell the cell, it's time to leave. Now that can happen when a cell becomes, for example, cancerous, develops mutations, or it's a natural part of the developmental process. And here is an incredible idea. Here's my hand, all right? Now notice distinct fingers. But when my hand was an embryonic hand, it was just one paddle of tissue. And then apoptosis knocked out the cells over here. It sculpted the form. So that's why it's B. Apoptosis involves the regulated activation of proteins in specific cells that lead to the death of those cells. Apoptosis, a very important developmental process. Okay, so hope everybody's doing okay. Hope everybody's getting what they came here for tonight. I'm going to look through the chat just to see if there's anything important. And hey, I'm going to go with you guys want you want me to answer a specific question? Give me a super chat. Why the heck not? That's super fun, or a super sticker. And someone whose name is kind of like fairy godmother is saying, will there be a recording? Yeah, these are all automatically recorded. So you can watch them live, as so many of you are, or you can watch it on replay. And it's just in the Science Music Videos channel, which you obviously know about under the Live tab. Okay, so I'm not seeing any questions. I've invited you to super chat, and Elan is asking, how in depth do I need to know photosynthesis? You need to know a lot about it. You need to know the light reactions, and you need to know the Calvin cycle. But like, for example, in the Calvin cycle, you don't need to know about like any of the names of any of the enzymes or any of the compounds. And that's it. And Mazzy, how much should we study if we start today? I think I just talked about that. A couple of hours a day. Couple of hours a day. And someone wants me to shout out Mr. Okawa from Oahu, Hawaii. Wow, that's exciting. Yeah, sure. Hey, Mr. Okawa, you're doing a great job. And ADPH. Okay, Amber, you mean NADPH, and that is a mobile electron carrier that plays an important role in photosynthesis. It basically provides reducing power to the Calvin cycle. It enables the Calvin cycle to convert carbon dioxide into carbohydrate. And I'm just going to direct you where to go. Go to learn-biology.com. You know, folks, I'm going to show you. Let's see. I'm going to learn-biology.com. You're not seeing this yet. But now I'm going to go like this. And I'm going to share this tab. Okay, so learn-biology.com, go to courses, go to the AP Bio video pathway, and then just scroll down to our menu, which is really big. Okay, there we are. And here, like photosynthesis, the Calvin cycle. So Amber, this is for you, the Calvin cycle. Do this tutorial, all right? So if you're like doing these questions and you're feeling like, oh my God, I don't know this stuff, on learn-biology.com, we got tutorials about every single topic. Let's go back to our questions. Okay, next question. Lactose digestion in E. coli begins with its hydrolysis by the enzyme beta-galactosidase. The gene encoding LAC-Z is part of a coordinately regulated operon containing other genes required for lactose utilization. Which of the following figures correctly depicts the interactions at the LAC operon when lactose is not being utilized? So here's what would actually be useful. I'm starting to be a little bit confused by the letters that are coming in. Can you please put a 6, then a dash, and then A, B, C, or D? All right, 6 dash. Let me see what the answer is. And I see answers from Mazzy, from Kareem. Anybody else? 6 dash. Okay, Snickers. Snickers, who gave me a super chat. My first one ever. I love some of your names. Okay, folks, when lactose is not being utilized, and the overwhelming favorite is D, and D is the answer. Okay, here's an operon. Got a regulatory gene, got a promoter, got an operon, got structural genes, right? That's the structure. That's what all these things do. When tryptophan is in E. coli's environment, what happens? I'm having some difficulty loading questions. Okay. Got a couple of problems here. So you know what I'm going to do? I am going to go to the next question. Not sure what happened here, but let's just jump right here. Sorry about that for that little glitch. All right. Simple cuboidal epithelial cells line the ducts of certain human exocrine glands. Various materials are transported in or out of the cells by diffusion. The formula for the surface area of a cube is 6 times s squared. And the formula for the volume of a cube is s cubed, where s equals the length of the side. Which of the following cube-shaped cells would be most efficient in removing waste by diffusion? Folks, this is A, this is B, this is C, this is D. Please put a 7 and then your answer, A, B, C, or D. A, B, C, or D. Send it, folks. 7 dash, A, B, C, or D. Okay, folks, people got it. And that makes me really happy because the answer is A. This is about surface area and volume. I hope my slides show when but basically there's A. And what do we got? So here's the important thing. It's a little bit counterintuitive. A large cell has more surface area than a small cell. But the ratio, the surface area to volume ratio, is much higher in small cells than it is in large cells. So the basic idea is that as an object gets larger, its amount of surface area relative to its volume decreases. In this example, the larger cell's surface area to volume ratio is 1/10 that of the smaller cells. This explains so much. It explains, first of all, why cells are small, because a large cell can't use diffusion to get the nutrients it needs in and out and to get waste out. And this explains so much of biology. It explains why we have things like gills. Why? Because all those thin flaps are great at diffusion because they increase surface area. It explains why elephants have huge ears. Why? Because huge ears are flat. A piece of paper is flat. It has a huge amount of surface area relative to its volume. Why the mitochondrial membrane is so intensely folded. Why? Lots of surface area for all those enzymes. And then it's why the villi of the intestine is so folded. So that's what we got. And it also explains why whales are big. Whales are big because by being big, they have relatively little surface area relative to their volume. Okay, so looking at the chat, how are we doing? Crystal is asking, is it always the smallest cube? If they're asking you about which organism has the most surface area relative to its volume, it's going to always be the smaller thing. But it always depends on what they're asking because, like, for example, if the question was which organism would have the most difficulty in a cold environment, in that case, the smallest thing would have the most difficulty. Now, that means it's the answer, but it's just the opposite. All I'm saying, Crystal, is you got to pay attention. You got to pay attention to the specific question, okay? All right, scanning the chat. Nothing in here. Yeah, and someone's asking, and it's Ashritha. You're asking if it asks which has the largest surface area. Well, the whale has more surface area than the mouse. There's no question about that. But they're almost always going to ask you about the ratio of surface area to volume. So again, the highest surface area to volume ratio, that's the smallest thing. The lowest surface area to ratio, that in general is going to be the largest thing. And that again is because of the math. Surface area is something, it's about squaring something, whereas volume is about cubing something. So volume always goes faster. Let's talk about DNA replication. When DNA replicates, each strand of the original DNA molecule is used as a template for the synthesis of a second complementary strand. Which of the following figures most accurately illustrates enzyme-mediated synthesis of new DNA at a replication fork? They're giving you very schematic diagrams of a very complex process. Please, when you're ready, please, when I say send, people are already doing it. All right, go ahead, send it. 8. What represents it most correctly? Okay. All right, I'm looking at a lot of D's coming in, and D is indeed the right answer. Here's what you're looking for. The direction of replication is going to be opposite on the two strands. So that means right away we can eliminate this one and this one. So A and B are out. In C, the problem is that on one strand, the replication is going to be fragmentary. and on the other strand, it's going to be continuous. And this stuff gets super confusing, but in the 5 to 3 direction, 5' to 3' direction, which is what's going on over here, replication will be continuous. In the 3' to 5' direction, it'll be discontinuous. It'll be Okazaki fragments, named after the Japanese scientist who discovered it. Here's DNA replication. It begins at an origin of replication, and that separates the strand, creating a replication fork. And then a whole team of enzymes comes in. So for example, this one at 2, that's DNA polymerase. That's the main enzyme that is connecting new nucleotides to the growing strand. Here's DNA polymerase as well. DNA polymerase here is moving away from the replication fork. DNA polymerase here is following the replication fork. This direction, 5 to 3, is going to be continuous. This direction is going to be discontinuous. And there's a whole lot of enzymes going on here. And I'm not going to go through all of them, but just this is kind of showing you. On this strand over here, L, we have continuous replication. DNA polymerase is following the replication fork. On this strand, the lagging strand, DNA polymerase synthesizes in the opposite direction from the replication fork. So Cocoa for Cocoa Puffs. This is all in Unit 6. So you want to go to Unit 6 on learn-biology.com. And I got a tutorial, I got a video about DNA replication. Jean is saying that she loves my glycolysis song. I love it too. Thank you. Glycolysis. Come on, sugar for the breakdown. I also have a song about DNA replication. It's kind of to a salsa beat. It hasn't been my most popular song ever, but it really does the job. And it's got all these great animations. So watch my DNA replication song. You'll really learn a lot. Scanning the chat. Unit 6 is treacherous. That's fantastic. Okay. Yes, Alon, that was a self-plug, but I'm doing a lot of self-plugging. Booper schmooper, no immune system is on the exam. If your teacher is teaching an up-to-date curriculum, then they didn't teach you about the nervous system, they didn't teach you about the immune system, and that's just fine. Those haven't been on the exam since 2019. Angela, the 5 to 3 thing is complicated. And please go on to learn-biology.com, do a tutorial, and let's go. Okay. Okay, a little ecology. The following is a food web for a meadow habitat that occupies 25.6 kilometers squared. The primary producer's biomass is uniformly distributed throughout the habitat and totals 1,500 kilograms per square kilometer. Developers have approved a project that will permanently reduce the primary producer's biomass by 50% and remove all rabbits and deer. Which of the following is the most likely result at the completion of the project? A, the biomass of the coyotes will be 6 kilograms, and the biomass of the hawks will be 0.5 kilograms. B, the biomass of the coyotes will be dramatically reduced. C, the coyotes will switch prey preferences and outcompete the hawks. D, there will be 50% fewer voles and 90% fewer hawks. Folks, I'd like you to put the number 9, a dash, and your answer to this question, please. Okay, so here's a great chance for you to use your process of elimination smarts. And really what you're trying to do is this. Remember that the question writers, they need to tell 3 lies and 1 truth. And so like here, the specificity of this over here, the biomass of the coyotes will be 6 kilograms and the biomass of the hawks will be 0.5 kilograms. Come on, there's no way that they could predict that to that level. So that you should just sort of like sniff that one out and say that can't be true. The biomass of the coyotes will be dramatically reduced. You're probably going to want to put a check mark next to that one and say, yeah, that one could be true. C, the coyotes will switch prey preferences and outcompete the hawks. I don't know, that doesn't sound right. And then D, there will be 50% fewer voles and 90% fewer hawks. Again, that one fails because it's too darn specific. Okay, so use process of elimination, and I see a really smart comment from Seriously Everything, and I'm actually going to reinforce that right now. Seriously Everything is talking about the 90% rule, and that's what this is right here. So this is your typical flow of energy in an energy pyramid. If the producers had 10,000 kilocalories of energy, then that same ecosystem would have only 1,000 kilocalories, 1/10 of the energy at the primary consumer level than you'd find at the producer. And in every level, it goes down by another 90%. Someone's saying, I thought it was the 10% rule. You can think of it in two ways. You lose 90%, or each higher trophic level has 10% of the energy of the one above it. So as long as you know the relationship, you'll be fine. Shows the amount of harvestable chemical energy in each trophic level. Each trophic level has 10% of the chemical energy of the level beneath it. What are the reasons for the 10% rule? One is energy lost to heat during any energy transformation. It's the second law of thermodynamics. And if you're like, what? Don't worry about it. This is biology, not physics. Second is that there's energy lost through metabolism. Like right now, you are radiating heat into the environment. So is every organism. And that heat that's lost to the environment, that's not available to, these organisms that would be consuming these organisms. Limits to harvest efficiency. The grasshoppers don't eat every little blade of grass and so on and so forth. And there are limits to what organisms can assimilate. In other words, they poop out a lot of stuff that they don't absorb. I have a song that's called the Food Chain Song that you should listen to. It's really a cute little song. Oh, a nuclear reaction happens deep inside the sun where hydrogen atoms are fused into helium. And it goes on, and it explains everything that you need to know about trophic levels. Any tips for FRQ studying? You know what, folks? I actually think that we should shift and we should do some FRQs, all right? And Seriously Everything is asking the difference between a decomposer and a detritivore. Decomposers are like fungi, and detritivores are probably also fungi. Detritus is the stuff that falls down, like leaves fall down. I do not think you need to worry about the difference between any of those. And I want to say to Alexa, my teacher hasn't given us any FRQs. Am I cooked? You're not cooked, but you have some work to do. You have some work to do. And the work that you have to do is this. I hope this is okay for a little self-promotion, but I'm going to share this tab over here. Okay, so we're back to learn-biology.com. See where it says exam review? Click on exam review. And then you see these AP Bio Enhanced FRQs. We're not going to do these now because you can do them on your own. I want to go over some published FRQs. But if you click, that's good. Okay, good. Good. I'm just going to randomly pick up an FRQ. Okay, so this one is about, I'm going to make it a little bigger. Hemophilia is an X-linked recessive disorder in which the ability of blood to clot is severely reduced. Rachel's brother has hemophilia A. Neither Rachel nor her parents show signs. If Rachel has a son with a man who is not a hemophiliac, calculate the probability that the son will have hemophilia A. Justify your response. And then there's another question. All that I'm saying is that like you would type in a response over here and then you'll get feedback. You'll get feedback. If you're stuck, you can get a hint, right? And you can also look at a sample answer. So you can just click I'm stuck, show the answer. And like here's the sample answer. And this is what we were asking you to do. And you'll notice that this is like the complexity of AP Bio questions. So, they're not actual AP Bio questions because those are copyrighted questions by the College Board. Anyhow, please spend your study time doing stuff. And the stuff that you got to do are FRQs and MCQs. Let's do some FRQs. Let's start with this I've made a bunch of videos about FRQs, and so I want to do ones that I haven't done videos about. So I'm scrolling down, and oh, you're not even seeing what I'm sharing. Now you are. So let's go to 2020. So I have videos about all of the 2024 FRQs that explain all of them, and you can find them on Science Music Videos. I even have a playlist about FRQs, but I haven't done much in the way of videos about 2023. So let's do this one. Okay, so this is okay. Got a message that my slideshow wasn't loading. That would have been catastrophic. So this is no. All right, I'm going to jump to the right question. Sorry, I want 2023, question 2. Here we are. Okay, so in this question, has something to do with mitochondria and CO2. We're going to do some bar graphing, and we are going to do environment genotype interaction. We're going to talk about non-nuclear inheritance. Oh, and I just want to let people know, I've got an FRQ guide. And just go to apbiosuccess.com, download the FRQ guide. So, question 2. Let's read it together. I'm going to read it out loud. Elevated levels of CO2 increase the rate of photosynthesis and growth in plants. Scientists studying the mechanisms involved in these increases examined a variety of species and found that when plants are exposed to elevated levels of CO2, there is an increase in the number of chloroplasts per cell. To investigate whether the elevated levels of CO2 have a similar effect on the number of mitochondria in plant cells, the scientists then selected 6 of these species to quantify the number of mitochondria per cell when the plants were exposed to both normal and elevated levels of CO2. Now, I just want to emphasize, I've taken this question and I've reformatted it, but this is the 2023 exams question 2. And you can get this from the College Board's website. So here they are. You've got 6 species, and you can do the following. Just take a look at the number of mitochondria per cell, 6 different species. And this is at normal CO2 levels. This is at elevated CO2 levels. And someone has a comment. Linda made a comment of use the practice test from the College Board. Yeah, sure, absolutely. They wouldn't display well here in this format, but everybody should be going to the College Board's website, downloading those FRQs, and spend a lot of time practicing with those FRQs. It's the best thing that you can do to prepare for the AP Bio exam. So Linda, good job. That's exactly true. So Ava, that's such a great question. And Shreyans, no, ban this man. It's not always a bar graph, all right? So let's take this question piece by piece. First of all, first question is describe the role of the inner mitochondrial membrane in cellular respiration. I'm going to try an experiment. I've never done this before. Here's what I'd like you to do. I would like you right now to type into the chat. Don't send it yet, but just type your answer into the chat. Your answer. What's the role of the inner mitochondrial membrane in cellular respiration? All right, don't send it. Don't send it. Just type your answer to question 2A while I take a sip of tea to soothe my increasingly hoarse throat. Okay. Everybody's writing their response. And please, I know I'm rushing you like crazy. Send it. What's the role of the inner mitochondrial membrane in cellular respiration? Go. Ilan, nice. Okay. 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 So here's the answer. The inner mitochondrial membrane contains the enzymes for the electron transport chain, proton pumps for creating a proton gradient, and ATP synthase, which produces most of cellular respiration's ATP through chemiosmosis. Now, if you look on the scoring guide for this question, I think you'll find a shorter answer. But this is the kind of thing that I would want my students to write, like this level of specificity, because what the graders are looking for, and I've been a grader, is they're looking for specificity. Want to just respond to a couple of things here. Let's see if I've got. Krebs cycle happens in the matrix. This is the electron transport chain over here. So everything in arrow 2, that's the electron transport chain. The electron transport chain includes proton pumps that are pumping protons from the matrix to the intermembrane space. That creates a gradient. The only way that these protons can escape is through a channel and an enzyme ATP synthase, which uses diffusion protons kinetic energy to make ATP from ADP and P. The mitochondrial electron transport chain uses electron energy for pumping protons from the mitochondrial matrix to the intermembrane space, increasing proton concentration in that place. The only way the protons can escape is through a channel and an enzyme ATP synthase, which uses diffusion protons kinetic energy to make ATP from ADP and P. Yeah, that's the electron transport chain song. The chorus really explains everything you need to know about this. So there we go. Guys, now you got to do some graphing, but we're not going to do graphing here, but we are going to talk about this a little bit. First of all, let's just talk about some of the issues. First issue, what kind of graph? We've got 6 different species. When you have different categories of data, you use a bar graph. When you have one continuous variable like temperature or pH or oxygen level or something like that, then you can do an XY graph. But this is bar graph. It's 6 different species. And here's the way to think about it. If you can't extrapolate between the 2 data points, then you have to use a bar graph. And you can't extrapolate between species 1 and 2. There's nothing in between those 2 species. They're different species. You're going to need to do error bars. And error bars show that this is a range of data, like your mean was 1.0, but the range of the data went up as high as 1.1 and as low as 0.9. So your graph needs to represent that. And you need to have good scaling and stuff like that. So here's your graph. First of all, we've got our 6 species. Here's how we're going to scale it. We're going to go up to 3 because our biggest data point is 2.4 over here. And we need to go 0.2 above that. So, and Candace is saying, when you see time, line graph, yeah, sure. So that's how you start. You scale your graph. And then we're going to do our first data point. All right. So species 1, it's 1.0 plus and minus 0.10. So that's how you do your error bar. And then we're going to do our second piece. So this was normal CO2. This is elevated CO2. And I'm 1.6 plus and minus 1.0 again. But now I need to differentiate between those two. So I'm going to create a key. Normal CO2 is going to be open. Elevated CO2 is going to have these diagonal lines. And then that's what you're going to do. So I'm seeing, wow, a lot of chat about the bar graph, but like do it the way I'm doing it. And I also explain this in several of these practice FRQs. So this is what your finished graph would look like. And Joseph, error bars are these things over here, and they're indicating that there was a spread of data. In other words, this is a mean. They took a whole bunch of measurements, and there was variability in the data, and the error bar represents the variability in the data. And someone just asked when this ends, and it ends in just 8 minutes. Or is that right? No, I'm going to go to 7:45, I think. All right. I don't know if you saw that. Based on the data in Table 1, describe the relationship between the level of CO2 and the average number of mitochondria per unit area of cell. So I keep on flashing that answer. I'm just going to give it away because I've given it away too many times. Here's the deal. As the level of CO2 goes up, the average number of mitochondria per cell increases. Yeah, so that's the deal. You see, there's no trend. These are all different species, and I got kind of freaked out for a second because I was looking at a trend. There's not. Basically, for each species, like here's species 1, normal, high CO2. Here's species 2, normal, high CO2. So in every single species, as the CO2 level goes up, the number of mitochondria increases. And also note this, there's no overlap between the error bars, which means that the difference is statistically significant. And that's super important. So I am going to make myself a little bit bigger for this demonstration that I love to do, like this. Okay. If the error bars are like this, that's a statistically significant difference. If the error bars are like this, if they overlap, then the difference is not statistically significant. Remember that, all right? Overlap, not statistically significant. No overlap, statistically significant. So back to this over here. At every level, in fact, I'm just going to go like this, make this even bigger. You can see how at every single level, there is no overlap between these error bars, okay? All right, next question. This is a tough question, and I got to get myself off the screen like this. Okay, good. The leaves of a particular plant species are typically green, but scientists notice a plant in which the leaves have white stripes. They determine that the stripes result from a mutation in mitochondrial DNA that interferes with the development of chloroplasts. The scientists cross plants using pollen from the plant with white striped leaves and ovules from a plant with green leaves. Your job is to predict the phenotypes of the leaves of offspring produced from this cross and provide reasoning to justify your prediction. So let's see what we got here. Okay, so I showed the answer, which means I'm just going to tell you. It's complicated. The scientists cross plants using pollen from the plant with the white striped leaves and ovules from a plant with green leaves. Predict the phenotype of the leaves of the offspring produced from this cross. Now, the stripes come from a mutation in mitochondrial DNA that interferes with the development of plants. So here's your prediction. All the leaves will be green. Why? It's because this is non-nuclear inheritance. And let's review that. In animals, all the mitochondria come from the egg. No mitochondria come from the sperm. The sperm has mitochondria, but they're in the tail. And during fertilization, the tail doesn't enter into the egg. So all that enters into the egg is the sperm nucleus, and that's how you go from haploid to diploid when that sperm nucleus comes in, and then we have two sets of DNA, and life begins. Now, in plants, all the mitochondria and the chloroplasts come from the egg. Right? So it's a similar thing, but plants have mitochondria and they have chloroplasts. They both have DNA, they both carry genes. So the pollen doesn't contribute mitochondria and the pollen doesn't contribute chloroplasts either. All the mitochondria and chloroplasts come from the ovule. No mitochondria or chloroplasts come from the pollen grain. So it's analogous to what happens in sperm. So what's the reasoning to justify your prediction? You got to say something like this: all the leaves will be green without stripes, and the justification is that the pollen is from the plant with the white striped leaves. The pollen doesn't carry the mitochondrial genes, therefore none of the mutated mitochondrial genes will pass to the ovum, and all the leaves will be green. Pretty complex stuff. And before I go on, I want to give a little inspirational message. Here's the inspirational message. I've graded these FRQs. I've graded FRQs that are like 9-point questions, and I've graded over 1,000 of them in one sitting at the AP Bio Read, where they fly all these teachers out to usually Kansas City to grade the AP Bio exam. Nobody, hardly, would get like 9 out of 9 on a question. Most people, unfortunately, are getting 2 out of 9, 3 out of 9. People who are getting 5 out of 9 are doing real well. So you have to study in a way so that you have something to say about most of these questions. But if you miss one part of an FRQ, it is not a cause for despair. It's just like, be kind to yourself, say, okay, I learned something, and then go on and keep learning. And study as many of these past FRQs as you can. As many as you can, because they won't repeat the exact FRQ, but some parts might be mighty similar. So go ahead and do your best and keep learning. That's the most important thing. I'm talking as if I'm ending this thing, but I'm not. I'm going to do the next question. There's a question. Yeah, I don't think I'm going to say anything in response to that. And Darini saying, do they repeat questions? They don't repeat questions, but themes repeat. And I would say that the best way to prepare for May 5th is to do as many of these past FRQs as possible. You got to watch out for one thing. If you do anything before 2019, then there might start to be content in there that's no longer on the exam, things like questions about the immune system and nervous system. But if you know that that's the case, then you'll be fine. So, and again, Darini, how should you prepare for the multiple choice questions? There's not a lot of public multiple choice questions. There are some in the course and exam description. There's a 2013 public practice exam that you can find just by like Googling 2013 public practice AP Bio exam. And that's about it. But again, I just want to emphasize that on learn-biology.com, and I'm not sharing that, but I'm going to share it now. On learn-biology.com, if you go to AP Bio exam review and you go to multiple choice review, you can see that I've got 475 multiple choice questions. They're not as long as the multiple choice questions that you'll get on the actual AP Bio exam, but they're good. And they'll give you great feedback and you'll really build your skills. I'm seeing something from Mihir. Mihir, like same thing. Get a subscription to learn-biology.com. It's not really a subscription because you're only going to be with us for 1 month. And just study every night. Just do all of our multiple choice questions. You'll get a lot of practice and that will move you up to the next level. So anyhow, hope that works for you. Let's go back to where we were. We were doing this. Here's another question. You can't see it. Now you can. And I'm going to make myself smaller. So I'm going to go like this. Bam. Okay. Explain why plants with the same genotype are able to differ in the structure and or number of organelles in response to changes in atmospheric levels of CO2. Let's do some writing, folks. I'd like people to write their response to this question. So put your response in the chat. Don't send it until I say send. So everybody's typing away their response to question 2D, number 3. And I'm seeing a lot of action in the chat. So maybe that action should quiet down for a little bit as people type their answer. And Big Man is saying, don't send. Way to go, Big Man. And I'm going to be quiet for just a moment to let you finish this task. I'm reading the question to myself. I'm thinking about how long it would take you to send it. And I'm also thinking about the fact that YouTube videos are very boring when they're silent for long periods of time. So I would say, send your answer now. And I just want to tell you guys how much I appreciate it. You know, this is a difficult, this is a difficult way to have like an interactive thing, but you guys are just doing such a great job answering these multiple choice questions, answering these FRQs. It's really, really fun. So here's the College Board's answer. This is an example of genotype environment interaction, during which changes in the environment, such as the level of CO2, affect the expression of different genes, such as those in mitochondria and chloroplasts, which alters the phenotype, such as the number of mitochondria and chloroplasts. So the big idea is genotype environment interaction. That, if you're unfamiliar with it, it's a topic in Unit 5, genotype environment interaction. And it's the really simple idea that everybody knows that genes don't determine everything. It's not just the seed, it's the soil that the seed gets planted in. Why do I have a picture of hydrangeas? The reason why I have a picture of hydrangeas is because they're kind of a famous example of genotype, phenotype, genotype environment interaction. As a gardener, you can make your hydrangeas express this pigment, which makes them purple, or this pigment, which makes them red. And you do that by controlling the acidity of the soil. So that is a well-known example of genotype environment interaction. Looking at the chat, phenotypic plasticity. Abdallah, I'm not that familiar with phenotypic plasticity. Like, I know what each of the words mean. And I think that genotype environment interaction leads to phenotype plasticity. Like, I think those two, but that might be a question that, like, you know, go to ChatGPT and say, like, what's the difference between genotype environment interaction and phenotype plasticity? And I think that the term that the College Board uses and what they'll be looking for more is genotype environment interaction. So first of all, I want to say thank you to all the people who entered in a response. So let's offer some takeaways. The first thing is you got to know cellular respiration, and you got to know mitochondrial structure and the electron transport chain. And this diagram is almost the level of complexity. It's a little bit too schematic, but like you need to know that like glucose goes into glycolysis, it creates NADH and ATP, and the result is 2 molecules of pyruvic acid, pyruvate. That pyruvate still has lots of energy that diffuses in through the mitochondrial membranes, and then the link reaction will snip a CO2 off of each pyruvate and also oxidize it to produce NADH. NADH is one of these electron carriers. It's like a battery that can get charged up, and it powers the electron transport chain, the last phase of glycolysis, which makes 7/8 of the ATP in the process. So note that during glycolysis, Link and Krebs, glucose and its byproducts are progressively oxidized as NAD plus and FAD are reduced. Reduced means given electrons, and they become NADH and FADH2. And then during the electron transport chain in chemiosmosis, these electron carriers get oxidized, and that powers ATP creation. And I actually sang the song about the mitochondrial electron transport chain. You should also know about environment phenotype interaction, and also know that like that is not some weird thing. That is the reality of life, that genes interact with the environment to produce phenotype. That's true of your very own life. You are where you are. You're taking AP Bio because of the environment that you got to be in. And, you know, taking AP Bio is a privilege. Here's a cool example. There are these rabbits. They're like Himalayan rabbits. Well, every cell in their body has the same genes, but notice that like their ears and the tip of their nose and their paws are dark. Why? Because those are far away from the body's core, and there's a cooler temperature, and different genes wind up getting expressed. That's another genotype environment interaction. Think about height and weight in humans. Think about skin color changes when you get a tan during the summer. That is a genotype environment interaction. There are reptiles that become male or female based on the temperature in the nest where they develop. If they develop in a cooler region of the nest, they might be male or female, and vice versa in a warmer region of the nest. Non-nuclear inheritance. Remember that not all the genes are in the nucleus. And so in non-nuclear inheritance, you'll get this kind of inheritance pattern where the mother passes on the genes to all of her offspring. The father, let's see if we have a dad. The dad, even if he inherits the condition, doesn't pass on the condition. Why? Because the dad doesn't pass on his mitochondria. Folks, take out your phones, take a picture of that QR code. If you want the best practice possible, go to learn-biology.com. You'll get enhanced practice FRQs that give you AI feedback. They're going to make you into a great FRQ writer, and there's still plenty of time to make it work. So folks, holy cow, we are going to come to an end now because my plan was to go to 7:45. I don't have time to do another FRQ. And I just want to tell you that I believe in you, and I think that, let me just check something that I might be able to show. Give me a sec. Just trying to figure something. Don't go away. Oh yeah, there we go. Okay. So let me share this. All right. So just want to let you know, if you want to do more of this, I do this. Some of them are paid. Some of them are free. So like, for example, tomorrow night, I have the ACE, the AP Bio Academy, and I'll show you how to go there in a sec. In fact, I'll put up a banner right now. You just go to AP Bio Success. No. Go here. apbiosuccess.com/exam review. And you can see that I have only 2 sessions left. I've already offered 3, and they're fantastic. You get to do this on Zoom, so it's much more interactive. And I've got one tomorrow night from 5 to 7 p.m. PST. Wednesday, I'll be back here live, 5 to 6:30 p.m. So I'm going to do it a little earlier for folks on the East Coast. And then Saturday, I've got a 4-hour Zoom review session, and I hope you can join me. And the way that you join me is by going to apbiosuccess.com/exam review. So I'll either be here on YouTube or I'll be on Zoom, and I hope that you can join me. Whatever you are doing, I hope you're using learn-biology.com, because that is what's going to get you to the level that you need to be at to crush it on the AP Bio exam. I want to tell you how much I admire you for being here on Monday night, how much I'm rooting for you, how I really, really want you to do the work that you need to work. Set the time aside every night, work for a couple of hours. I've got all the resources set up for you on learn-biology.com. I'll be back here on Wednesday night. And boy, that might be the last one that I'm going to do this season. So I hope I see you. And guys, thank you very much. And Glitch is saying this is a pre-recorded video. Are you kidding me? This is live. And you know that it's live because I just responded to your comment. So anyhow, there we go. And I'm looking at the chat, and I think that's it. So folks, I'd really appreciate it if you put some thanks in the chat. Just say thank you, signing off from wherever you're signing off from, because I'm signing off from Berkeley, California, the home of learn-biology.com and the Science Music Videos channel. Go to Science Music Videos. I've got all these unit review videos that are really going to help you. It's been great being here with you tonight. Everybody stay safe, study hard. Good luck on May 5th. Everybody be well. Thank you.